Hey guys, we are in lesson 8.3, cell transport, and I'm just going to go over this with you. All right, if you are in, let me back out into the class so we can see how we got here. We're in our course. You're clicking on the materials section, and in the Unit 3 folder, that's where you're going to find the slideshow. Click on that. Unit 3 resources, and the slideshow is right there, the lesson slideshow. If you're taking notes, there's your interactive notebook, and then at the conclusion of today's lesson, you will be doing the 8.3 reading quiz. So if you are following along with student completion, you should be up to the 8.3 reading quiz at this point. You can see February 16th, February 17th, and then February 18th. 8.3 is right there. Looking ahead, today is Thursday, February 18th. We are in lesson 8.3. And as you know, next week we will be wrapping up Unit 3. You've got some study islands coming down the pike. You've got your vocabulary packet and your vocabulary quiz due, and you'll have your exam next Friday. So you do have a lot of high point value assignments coming up next week. It would be in your interest to get started on those. So don't forget about all of the big stuff that's coming due next week. All right, that being said, let me get into the lesson for today, which is in the Unit 3 resources folder in the Lesson 3 slideshow. We are in Lesson 8.3, Cell Transport. Slide number 75 here has a T-chart that would be beneficial for you to either write down on paper or keep in your virtual notebook. And here is the link to your virtual notebook. This will be a short answer question on the test, and it's a T-chart that compares the different types of cellular transport, active transport and passive transport. Cell transport really has everything to do with how materials get into and out of the cell. This picture here is a cross section of the cell membrane. We talked about the cell membrane a little bit in Unit 2 when we talked about lipids. And we said one of the things that's great about lipids, in addition to it being long-term energy storage, it also provides a waterproof covering for biological membranes. And that's where this comes into play. If we look at this cell membrane here, you see it's composed of a phospholipid bilayer, which means two layers of phospholipids. This would be the inside of the cell, and then this would be the outside of the cell, and this cell membrane encapsulates the entire thing, like the balloon part of the water balloon. In the cell membrane, you have these little proteins and channels embedded because they decide what can move into the cell and what has to stay out of the cell. And we call that selective permeability. So that means it selects. You see selective permeability. I believe that's a vocabulary term. It selects what materials can get in and what materials can't, who has to stay out, because you don't want everything to just be able to get into the cell. So there's your cell membrane. The next two slides are just some more images of that. Like we talked about, there's your phospholipid bilayer. We talked about that in Unit 2. Here's another picture of our cell membrane with the materials that are in the cell membrane, cholesterol, proteins, and carbohydrates. A little bit more about selective permeability. It allows some things to get in, but not everything. It's selective about it. And when we talk about selective permeability, we have to explain that in terms of concentration gradient. You know, when something is a high concentration, that means there's a lot of it. And when something's low concentration, that means there's less of it. So when materials follow a concentration gradient. That means they go from an area of high concentration to low concentration until evenly distributed. It's like if you put a little bit of food coloring in a cup and you waited for like a couple hours, even if you didn't stir it, eventually that food coloring would diffuse throughout the cup of water until evenly distributed. And so that's what we talk about when we talk about concentration gradient. It means materials moving from high concentration to low concentration in a certain direction. When we talk about materials moving across the cell membrane, there's two types of transport. So looking back at your T-chart that you want to be writing these notes down on or typing them in your notebook. The first type of transport is called passive transport. And passive transport does not require any energy from the cell. It relies on the principles of physics. If you sprayed some Axe cologne in my classroom, I'd be really mad. But that cologne would travel from an area of high concentration, where it was sprayed, throughout the whole room, and we'd all be smelling it and we'd all be mad until it's evenly distributed. So it travels throughout the room until evenly distributed. And it doesn't require any energy to do that. It's passive transport. It happens without energy. The types of passive transport that cells rely on are diffusion, osmosis, and facilitated diffusion. Don't get scared about the word facilitated diffusion or like make it into something bigger than it is, because it's still passive transport. Your diffusion is something we've all come in contact with. It's the movement of molecules from an area of high concentration to low concentration until evenly distributed. Osmosis is passive transport that only deals with water. And facilitated diffusion is passive transport, but it relies on one of these little channels that's inside the cell membrane to help molecules move across. And that's what the word facilitated means. The word facilitated means to help. So it's going to move those molecules across the cell membrane. All right, so those are your three types of passive transport. When we're talking about osmosis, there's three, let's call them flavors of osmosis. There's isotonic, hypotonic, and hypertonic. All right, these three slides go into pretty good detail with those. I'm not going to read them to you. I'm going to jump down to, you can read them, please read them. I'm going to jump down to slide number 87 here, though. And these images, I really think, help to get us to understand the relationship to solute concentration in water when in regards to the cell. If we are in a isotonic solution, that's the just right solution. That's when the concentration on the inside and outside of the cell is the same. So you have these solutes, and they could just be different types of molecules. You have these solutes moving evenly into and out of the cell. Okay, that's isotonic. A hypertonic solution will create a cell that starts to shrivel up. It would be like if you took a cell and put it in some salt water, all of the water is going to get sucked out of the cell. It's going to exit the cell and it's going to shrivel up. So in a hypertonic solution, your cells will shrivel up. And then in a hypotonic solution, this is where we have the opposite of hypertonic. Your cells have a lot of solute on the inside. So they're trying to even out. They want to be isotonic, so they suck in a lot of water and they could potentially burst, or it's called lyse, Lyse. They could burst open. So those are the three states that a cell could find itself in when we think about the relationship between solutes and water in the cell. All right, so those are our three types of passive transport: diffusion, facilitated diffusion, and osmosis. Next up, we have active transport. And as the name would suggest, this does require energy from the cell. And we're going to be talking about that cellular energy during all of Unit 4. Cellular energy is going to be moving the molecules against the concentration gradient. So you can picture me in the classroom trying to fan that Axe body spray away from me. I've got a folder and I'm fanning away the Axe because I don't want to smell it. And that requires a lot of energy. So that's active transport. The three types of active transport that take place in cells are endocytosis, phagocytosis, and exocytosis. These are easy to remember if we think about our prefixes. Endo, the en, is going to make you think of the word enter, which means it's taking in large molecules. Phagocytosis is endocytosis that just refers to eating. It's when unicellular organisms use this active transport to absorb their food. They eat this way. And then exocytosis is when large molecules exit the cell. All right, so those are your two types of, those are your active transports. All right, active transport requires energy. We're going to talk more about the energy for cells. It's called ATP, or adenosine triphosphate. We're going to talk more about that in Unit 4. And the next few slides just go into that in a little bit more detail. Your endocytosis, phagocytosis, and exocytosis. The EX makes you think of the word exit. We're going to stop right here for a second. In your notebook, you're just going to go back and double check all of these slides and make sure you've written down the information related to passive transport and the information related to active transport. You can double check your work with slide number 94, because this is where I wrote down in my T-chart passive transport, diffusion, facilitated diffusion, and osmosis. Active transport, endocytosis, exocytosis, phagocytosis, and the sodium potassium pump. Now, I didn't get a chance to talk about the sodium potassium pump. Let me go back to this slide here. If we're looking at the cell membrane, you see these integral proteins, which are part of facilitated diffusion, but there's another embedded molecule in the cell membrane, and it is called an ion channel. The ion channel moves only sodium and potassium ions across the cell membrane. And the sodium and potassium ion gradient is really important. They've actually discovered that that's what the imbalance of that is what happens when you get a concussion. They used to think it was kind of like a brain bruise, but what's actually happened is the trauma of your brain injury causes these ion channels to stop working. So, where your body typically is pumping ions against a concentration gradient using energy, when you get a concussion, your brain cells stop doing that. And that's actually the damage that's caused by a concussion. It's damage to your ion channels. So that's kind of interesting. There's still a lot of research taking place with brain injury, and how it's more of an active transport issue than it is like an actual bruise. So double check your T-chart. Make sure it looks like mine. This will be a short answer question on your test. You do not need to submit this anywhere. Your assignment for today, after you watch the video, is to read Pearson 8.3 and complete the 8.3 reading quiz in Schoology. If I did a terrible job explaining this, or you got offended by my dislike for Axe body spray, you can watch the Amoeba Sisters. They probably do a better job. But if you're still confused, if you want a little bit more support, or if you're just tired of listening to me at this point in the school year, go ahead and watch the Amoeba Sisters. You know they're short videos. You know they do a good job of keeping things interesting. And as always, your homework is always to work on your vocabulary packet. And if you're working ahead, go ahead and do a choice board activity for extra credit. It's a snow day. We have nowhere to be. So go ahead and do an extra credit project and email that to me. I hope you're well. I hope you have a good day, and I will talk to you later. Thank you.